Today we are going to discuss about structure of MHC and microbial biotransformation reactions, which is coming under the sixth syllabus, sixth sem BPharm of biopharmaceutical biotechnology. So coming to MHC or major histocompatibility complex. So MHC means it's a group of genes that code for proteins that usually present on the surfaces of cells that represent antigen to the T cells for intracellular recognition and discriminate between self and non-self. So we know that whenever any antigen or any foreign materials when enter into our body, we know that two types of cells will be get activated, either B cells or T cells. So the first cell that will get activated is always the B cells. But in some cases, due to some antigens, when it enters into our body, T cells also can be get activated. So if B cells are activated, the B cells can recognize the antigens by its own. They will come and bind with the antigen and they can produce antibodies and finally they will kill the antigen. But in case of T cells, the T cells has no ability to identify or recognize the antigen. They are not able to recognize or identify the antigen of their own. So that in that case, in our body, whenever an antigen is entered, this antigen will come and bind with some protein called as MHC. So MHC is a protein that will come and bind with the antigen. Once this antigen is bounded with the MHC, now these T cells can come and attack the antigen. Okay, that is the main function or main purpose of MHC in our body. Okay, MHC means these are a group of genes or these are a group of proteins which helps to recognize the antigen to the T cells. So T cells in ഒരിക്കലും ആന്റിജനെ ഐഡന്റിഫൈ ചെയ്യാനുള്ള എബിലിറ്റി ഇല്ല. സോ ഒരു ആന്റിജൻ ബോഡിക്കകത്ത് കയറി കഴിഞ്ഞാൽ ഈ ആന്റിജനെ T സെൽസിനെ റെക്കഗ്നൈസ് ചെയ്ത് കൊടുക്കാൻ വേണ്ടിയിട്ട് ഹെൽപ്പ് ചെയ്യുന്ന പ്രോട്ടീൻസ് ആണ് ആക്ച്വലി MHC. ഓക്കെ, ആൻഡ് ദിസ് MHC is also having another function, that is tissue allo recognition. Okay, at the time of organ transplantation and also this MHC is playing an important factor. And in human beings, this complex is called as HLA. So in human beings, we call this MHC as a HLA system. And mainly two types of MHC proteins are there, MHC1 and MHC2. Okay. So here in this picture, you can see the B cell receptors can recognize the antigen on their own, whereas the T cells cannot recognize the antigen by their own. They require a mediation through MHC. That means once the antigen is entered in our body, this MHC will come and bind. Here you can see this MHC protein will come and bind with the antigen. Once this MHC protein is come and bind with the antigen, then the T cells will come and attack. Otherwise, the T cells cannot recognize this antigen. So that is the main purpose. So we said that this MHC are a group of proteins usually present in some cells. So these MHCs are usually present in antigen presenting cells. Okay, we cannot see this MHC in every cells. These MHC proteins are usually present only in the antigen presenting cells. So now we are going to discuss what is antigen presenting cells. Antigen presenting cells are a group of immune cells that mediate the cellular immune response by processing and presenting antigens for recognition by certain lymphocytes such as T cells. Okay, so antigen presenting cells means these are the cells which help the T cells to recognize the antigen. So that means these cells, in the inside of these cells, you can see MHC. Here you see this is the antigen presenting cell. Inside the antigen presenting cell, MHCs will be present. So once this antigen come inside this antigen presenting cell, this MHC will bind with the antigen and the antigen MHC complex then will get projected outside the APC. That means these APC cells or antigen presenting cells will present the antigen towards the T cells. Okay, so examples of those cells are dendritic cells, macrophages, Langerhans cells, and B cells. B cells are also a type of APC cells. Coming to the definition, so major histocompatibility complex is a membrane-attached protein which work on recognition of antigen between self and non-self body against antigen presentation. So I think it's now clear. So antigens, once it enter into the body, if T cells has to recognize this antigen, this this antigen should be binded or bounded with the MHC. Then only this T cells can come and attack this antigen. Otherwise, it cannot recognize the antigen. Okay, so in order to recognize or in order to present the antigen towards the T cells, this MHC will be acting as a mediator. Okay. So major histocompatibility complex is a set of surface proteins located on the cell membrane of the antigen presenting cells or nucleated cells. It's a play important role to identify the antigen between self and non-self body, intracellular recognition, and responsible for antigen presentation. And from the name, you see that major histo. Why? Histo means tissue. The meaning of histo means tissue, and compatibility refers to living together. So I said that it is not a single protein, it is a group of protein. So a group of protein living together in the cells or tissues. That is the meaning of major histocompatibility complex. Okay. And these molecules always recognize only to T lymphocytes. And T lymphocytes, you know that two types of T cells are there, T helper cells and cytotoxic cells. So if we want to recognize the antigen to T helper cells, then MHC2 molecules will be acting as a mediator. And if we want to present the antigen towards T cytotoxic cells, then MHC1 molecule will be acting as a mediator. That's that. That means if an antigen is enter into our body, if the T helper cells want to come and bind with this antigen, then who will work? This MHC2 molecules will be working as a mediator. That means MHC2 molecule will come and bind with the antigen and it will get presented towards T helper cells. But in case of if the antigen has to be recognized with the T cytotoxic cells, then which MHC will act as the mediator? MHC1 only will be acting as the mediator. So that difference you should be understand. Then classification. So we said that only about class 1 and class 2, not only two classes. There are 4 classes of MHCs, class 1, class 2, class 3 and class 4. But for in case of immunity and all, class 1 and class 2 is only important. Class 3 and class 4 not that much important. They are not playing any role in the body. So coming to the class 1 MHCs. So class 1 MHC molecules, the molecular weight is 45 kilodalton. And they are present on the surface all the nucleated cells except nervous tissue and platelets. You cannot find this class 1 MHC. So as I already said, class 1 MHC molecules will present the antigen to TC cells. TC cell means T cytotoxic cells. Okay. So T cytotoxic cells mainly consist of CD8 protein. So these CD8 proteins will be attached with this antigen and they will bring about cell-mediated immunity. Then coming to the structure of class 1 MHC molecules, it mainly consists of polypeptide chains. So since MHC is a protein, polypeptide chains are usually present. Two types of chains are there, alpha chain and beta 2 microglobulin chain. So just I will okay, so this is the structure as I already said. So alpha 1, alpha 2 and alpha 3. So this blue, green, these three globules it is the alpha chain and this red color, this globule is the beta chain. So alpha chain is divided into alpha 1, alpha 2 and alpha 3. And in between you can see disulfide bonds will be present and beta 2 chains are also consisting of what? disulfide bonds, and this alpha chains will be having a transmembrane protein. Okay, this coiled type, this coil is called as transmembrane protein. So this transmembrane protein is extending towards the cell membrane. Okay, so this one, we know that this MHC will be present or any antigen presenting cells. So the transmembrane proteins inside the MHC will come and extending towards the membrane of this antigen presenting cells. Okay, and once the antigen get internalized into the cell, this antigen will be come and binding with this portion. So this portion is known as peptide or antigen binding group. And so whenever the antigen come inside the cell, this antigen will come and bind with this portion of this MHC, and this portion will, and after that MHC will come outside this cell and will get presented towards the cytotoxic cells. Okay. Next is class 2 MHC molecules. Class 2 MHC molecules are also present in different types of antigen presenting cells, and it will bind with the CD4 adhesion molecules of T helper cells. So these class 2 MHC molecules always recognize or represent this antigen towards T helper cells. And it also consists of two polypeptide chains, namely alpha chain and beta chain. And if you are checking the structure, we said that it is having two chains. Alpha chain is 33 kilodalton and beta chain is 28 kilodalton chain. And this alpha chain is again classified into alpha 1 and alpha 2. And beta chain is again classified into beta 1 and beta 2. Beta 2 and alpha 2, both these chains will be having transmembrane domains anchoring towards MHC. Okay, so this is the class 2 molecules. So in class 1 molecules, we had only alpha chain and beta chain. Alpha chain was three globules were there: alpha 1, alpha 2, alpha 3, and beta 1. But in case of class 2 MHC molecules, here you can see this blue portion is alpha 1 and this one is the alpha 2, and the right side chain is known as the beta 1 and the red portion is called as the beta 2. In both the chains you can find the transmembrane proteins. Transmembrane proteins mean these are the coiled proteins. So these coiled proteins are anchored towards the cell membrane by the two chains. But in case of MHC1 molecule, only the alpha chain was having this transmembrane protein. But in case of class 2 molecules, both the alpha and beta chains are having this transmembrane protein. So that is the another difference. Then coming to the antigen will always come and bind with this portion. So this portion is known as peptide binding group. Okay. So that's all about the structure of MHC. So class 1, class 2 we already completed. Then class 3 molecules are usually present in a complement proteins such as C2, C4 and etc. And class 4 MHC molecules are also not that much important. They are usually present on the T cells of leukemia or immature thymocytes we can see. Then we said that the MHCs which is present in human beings are named as another term, that is HLA. Okay. അപ്പോൾ നമ്മുടെ ഹ്യൂമൻസിലുള്ള MHC നെ പറയുന്ന പേരാണ് HLA എന്നാണ് നമ്മൾ വിളിക്കാറ്, ഹ്യൂമൻ ലീക്കോസൈറ്റ് ആന്റിജൻ. Okay. HLA is the MHC molecule present in human beings and they are presented in all the nucleated cells, and the main function of this HLA antigen is to graft rejection in case of adaptive immunity, defense against infection, and sometimes it is expressed in cancer cell destruction, autoimmune disorders, and certain complements. Okay. Then coming to the functions of MHC molecules. MHC molecules are loaded with a sample peptide fragment derived from the degradation of proteins. This peptide is the mirror image of the proteins present inside the cell. MHC molecules contain self as well as non-self antigens. They bring about defense against infections and diseases. They can mediate autoimmune disorders and responsible for smell of an individual. So each individual will be having a unique smell. So the unique smell of an individual is mainly responsible due to the presence of MHC molecules. So that is also another important thing about MHC molecules. So in nutshell, what is MHC? MHC means these are a group of proteins present in certain antigen presenting cells. So what is the function of this MHC? These MHC molecules will come and bind with the antigen. Once this MHC is binded with the antigen, then the T cells can come and destruct the antigen. Unless what happens, if these antigens are not binded with the MHC molecules, the T cells are not able to recognize the antigen. നമ്മൾ പറഞ്ഞു ബി സെൽസിന് ആന്റിജൻ ഒരു ബോഡിക്കകത്ത് എൻ്റർ ചെയ്ത് കഴിഞ്ഞാൽ ബി സെൽസിന് അതിനെ പെട്ടെന്ന് ആന്റിജൻ ആണെന്ന് ഐഡന്റിഫൈ ചെയ്യാനും ബി സെൽസ് ആന്റിജന്റെ അടുത്തേക്ക് വന്ന് അതുമായിട്ടൊരു കോംപ്ലക്സ് ഫോം ചെയ്ത് ആന്റിജനെ ഡിസ്ട്രോയ് ചെയ്യാനുള്ള ഒരു എബിലിറ്റി ഉണ്ട്. വേറെ ടി സെൽസിന് ഒരു ആന്റിജൻ, ഒരു ഫോറിൻ ബോഡി നമ്മുടെ ബോഡിക്കകത്ത് എൻ്റർ ചെയ്ത് കഴിഞ്ഞാൽ അതൊരു ഫോറിൻ ബോഡി ആണ് എന്ന് മനസ്സിലാക്കാനുള്ള കഴിവില്ല. അപ്പൊ എന്ത് ചെയ്യും? ഈ ആന്റിജനെ നമ്മൾ ഈ പറയുന്ന ആന്റിജൻ പ്രസന്റിങ് സെൽസ് എല്ലാം സെൽസിന്റെ അകത്തേക്ക് ചെന്നിട്ട് ഈ MHC മോളിക്യൂളായിട്ട് ബൈൻഡ് ചെയ്യും. MHC മോളിക്യൂളായിട്ട് ബൈൻഡ് ചെയ്താൽ മാത്രമാണ് നമ്മുടെ ടി സെൽസിന് ഇതൊരു ആന്റിജൻ ആണെന്ന് മനസ്സിലാക്കാനുള്ള ഒരു കഴിവുണ്ടാവുള്ളൂ. സോ MHC ആയിട്ട് ആന്റിജൻ ബൈൻഡ് ചെയ്ത് കഴിഞ്ഞാൽ ടി സെൽസ് ആക്ടിവേറ്റ് ആയിട്ടിട്ട് ടി സെൽസ് വന്ന് ഈ ആന്റിജനെ കില്ല് ചെയ്യാൻ തുടങ്ങും. Okay, so that is the main function of MHC molecules. Be clear, MHCs are group of proteins present in certain cells. So the main function is in order to bind with the antigen and present the antigen towards the T cells. Okay, so that's all about MHC molecules. Next, we are going to discuss about microbial biotransformation reaction. So we know that for any reactions, in case of any reactions, what is happening? A substrate is converted into the product. Okay. So reactions are different types. Biological reactions are also there. Chemical reactions are also there. In chemical reactions, what we are doing? We are taking a substrate. We are adding some chemical reagents, some catalyst, some organic solvents, some things that we are added. Then we are giving some reaction time. Then you can see a product will be obtained in a laboratory, in chemistry lab, and we are performing those chemical reactions. Same like that, there are biological reactions are also there. So in biological reactions, we are not using chemicals. Okay, chemical reagents, catalysts, nothing we will be using. We are taking the substrate, we are adding some microorganisms or biological cells, and in the presence of these biological cells, the substrate is converted into a product. So that type of reactions are known as microbial biotransformation. Okay. So microbial biotransformation is a biological process in which organic compounds are modified into a reversible product. And these reactions are catalyzed by purified enzymes which is present in microbial cells or pure cultures of microorganisms. And these types of reactions are routinely used in the production of steroids, antibiotics, vitamins, prostaglandins, citric acid, and etc. Okay, so in order to carry out this biotransformation reactions, first of all, we require microorganism. So I already said that in order to convert a substrate into a product in microbial biotransformation reaction, we are using microorganisms. So microorganism is one of the important requirement for a microbial biotransformation reaction. So first step is we have to grow the microorganisms in a large fermenters. After the microorganisms are grown, we will be adding the substrate. What is the substrate? Substrate is the substance that has to be converted into a product. Okay, once first step is growing the microorganisms in a large fermenter. The next step is we are adding a substrate to the fermenter. Then third step, we are giving some incubation time. That is 24 hours, 48 hours, or 7 days. Like that, we know, same like fermentation process, we will be giving some incubation time. So during this incubation time, what happens? Due to the microbial, due to the presence of this microorganism, the substrate will be slowly converted into the product. Fourth step is we are extracting that product. Finally, we are purifying the product. Okay, this is the main steps involved in microbial bioconversion. So what is the advantages? So we said that microbial biotransformation is not a chemical process, it is a biological process. So what there are some advantages over chemical reaction. First one, it is easy. And we know that chemical reactions are not that much easy to perform, but the biological biotransformation reactions are easy because we are using biological approaches. And in case of chemical reactions, we required several solvents and organic catalysts. Okay, so that will be not much safer and it will be causing a lot of toxicities, then pollutions, etc. Then third advantage is that, and we in chemical reactions we can use organic catalyst, but in biotransformation reactions we are not using organic catalyst. Instead, we are using a biological catalyst. That means enzymes. Enzymes are the biological catalysts. They are always efficient than chemical catalyst. Then biotransformation reactions usually takes place under mild condition. It has reaction specificity, regiospecificity, and stereospecificity. And they can show catalytic activity both in aqueous and organic solvents. Okay. Then coming to disadvantage. One of the main disadvantage is that the expenditure is very high. Okay. In order to develop a biotransformation process and product isolation, the cost is very high. Reaction time is also rather longer, and sometimes the product concentration or yield will be, in some reactions it will be quite low, and stability is also sometimes limited. Okay, next is biotransformation designing. Okay, so in order to design any biotransformation reaction, three things we should be clear. First of all, how to select the microorganism, because microorganism is the vital part of this reaction. The second thing that the requirement is the condition, how the processing condition. The third thing, which is very important, is the recovery of the product. So these are the three things that is a vital part of biotransformation reaction. Okay, so coming to the first part, that is selection of microorganism. So a wide variety of microorganisms we can use. Okay, such as growing cells can be used, resting cells can be used, immobilized cells can be used, immobilized enzymes, cell-free extracts. These are the organisms we can use for biotransformation. So what is growing cells? So how we will use growing cells? Growing cells means these are the cells which will undergo vigorous growing when incubated with some cultural media. Okay, so in this process, the desired cells are cultivated in a suitable medium, and after 6 to 24 hours, if we know that the cell growth will occur. Once the cell growth is occurred, a concentrated substrate is added. Sometimes we can add some emulsifiers such as tween or water miscible solvents can be added, and we are giving some incubation time. In the time, substrate will be converted into the product, and this can be monitored by chromatographic or spectroscopic methods. Once the product formation is optimum, we can terminate the reaction. So this is the biotransformation reaction that takes place by using growing cells. Then non-growing cells or resting cells. The non-growing or resting cells are usually preferred in following cases. If the concentration of substrate is very high, if the concentration of substrate is very high, we cannot use growing cells, because growing cells will in the presence of high substrate concentration, the growing cells will stop their growth. Therefore, in that case, we are using resting cells. Resting cells means these are the microbial cells that will not grow. ഓക്കെ, ഗ്രോ ചെയ്യത്തില്ല. അതിങ്ങനെ ഒരു സ്റ്റേഷനറി ഫേസിൽ, ഒരു റെസ്റ്റിംഗ് ഫേസിൽ ഇരിക്കുന്ന സെൽസ് ആണ് റെസ്റ്റിംഗ് സെൽസ് എന്ന് പറയുന്നത്. ആ സെൽസും നമുക്ക് ബയോ ട്രാൻസ്ഫോർമേഷൻ റിയാക്ഷന് വേണ്ടിയിട്ട് യൂസ് ചെയ്യാം. Okay, and these cells can be washed and used, and thus there will be no contaminating substances. അപ്പോൾ growing cells, we are comparing growing cells and resting cells. Growing cells we cannot washed and used. Wash ചെയ്തിട്ട് പിന്നെ യൂസ് ചെയ്യാൻ പറ്റത്തില്ല. Whereas resting cells can be washed and used again. Contaminations will not be there. Conversion efficiency is very high. Comparing to growing cells, in order to get a high product yield, we can use resting cells. Then, in order to optimize the biotransformation reaction, resting cells are more helpful. Product isolation is also very easy with the resting cells. Then, immobilized cells. So, the third category of cells that can be used as microorganisms in biotransformation reaction, that is immobilized cells. Immobilized cells means these are the microbial cells that are binded with some carriers. Okay, that means they are live cells, but they cannot be moved. That means their movement is restricted or immobilized by binding with certain carriers. So what is the main purpose of these immobilized cells? Means we can reuse the cells for a number of times. Same microbial cells can be used for a lot of reactions. So that type of cells are known as immobilized cells. Single reactions or multi-stage reactions, we can use these immobilized cells, especially L-alanine, aspartic acid, malic acid. These are in these production of these compounds, we can use the same cells. Okay, like that immobilized cells, we can use immobilized enzymes also for the production of products. Then once the products are formed inside the fermenter, the product can be extracted and recovered by techniques like precipitation, extraction with the solvents. Then if it is a volatile product, we can use distillation, etc. So that's all about the biotransformation reaction, their steps, designing, etc. Next, we are going to discuss about types of biotransformation reaction. So actually this, in case of this biotransformation reaction, we are not using chemicals, but the reaction is always the same reactions only: oxidation, reduction, hydrolysis, esterification, halogenation, decarboxylation. These are the types of reactions that is taking place inside the biotransformation reactions also. But instead of chemicals, we are using microorganisms. In the presence of microorganisms, these types of reactions are happening. Okay. Okay. N-dodecyl benzene. Okay, this is N-dodecyl benzene. So this N-dodecyl benzene can be converted into phenylacetic acid with the help of Nocardia species. Okay, what happened here? The 12th carbon side chain gets oxidized to methane carboxylic acid in the presence. So C12H25 is converted into CH2COH. A phenylacetic acid is formed. Okay. Then epoxidation reaction. So 1,7-octadiene can be converted into 7,8-epoxyl-1-octane in the presence. Epoxides means, so in these are called as epoxides. Okay, here you can see that in the presence of Pseudomonas oleiferanes, what happens? C7 position, and in between C7 and C8 position, what happened? 1,7-octadiene will be formed. Okay. Next is oxidative cleavage of aromatic rings. So see, this is naphthalene. Naphthalene is converted into salicylic acid, not due to some chemicals or any catalyst. Here we are only used a biological organism, that is Corynebacterium. Okay, in the presence of Corynebacterium, what happens? An aromatic ring of naphthalene cleaved to form salicylic acid. Next is oxidation of heterofunctional moieties. Here you can see 2-amino 4-alkyl imidazole. In the presence of Streptomyces, converted into 2-nitro 4-alkyl imidazole. An amido group is This is the NH2 group is converted into a NO2 group in the presence of Streptomyces species. Okay. Next, coming to reduction reactions. So in the first reaction, an aldehyde group is converted into an alcohol group. You see, benzaldehyde, CHO group converted into benzyl alcohol, C2OH, in the presence of Saccharomyces cerevisiae. Okay, here what reaction occurred? Reduction of an aldehyde group. Aldehyde group is converted into alcohol. Then here you can see reduction of heterofunction. The nitro group, nitropentachlorobenzene is converted into pentachloroaniline. NO2 group is reduced into NH2 group in the presence of Saccharomyces orophyciens. Then reduction of carbon-carbon bonds. Okay, this alpha beta unsaturated carboxylic acid is reduced in the presence of Clostridium bacteria. Coming to hydrolytic reactions. Okay, hydrolysis, NCN. NCN is an alkyl nitrate group. It is hydrolyzed to form N-alkyl amide and aliphatic carboxylic acid in the presence of Bacterium. Then hydrolysis of esters of carboxylic acid. Here also you can see that the esters of carboxylic acid, Mycobacterium fei, aids the hydrolysis of D1-methyl laurate into methanol. D1-methyl laurate is converted into menthol. Just see these reactions only. Study this reaction and study the organisms. So in nutshell, we can say that a lot of reactions can be taken place in biotransformation reactions. So in case of oxidation, tryptophan can be converted into phytoxytryptophan by using Bacillus subtilis. Naphthalene can be converted into salicylic acid by Corynebacterium species. In case of reduction reaction, benzaldehyde to benzyl alcohol by Saccharomyces cerevisiae. Then nitropentachlorobenzol can be converted into pentachloroaniline by Streptomyces orophyciens. In case of hydrolysis, anhydrotetracycline can be converted into tetracycline by Streptomyces orophyciens. Menthol laurate to menthol divided by Mycobacterium feli. Then another type of reactions are condensation reactions. A lot of reactions. In the first slide, we already said that 12, 13 reactions are there. Okay. Streptomycin can be converted into streptomycin phosphate by using Streptomyces rhesus. Okay. So these are all the biotransformation reactions. Then next is biotransformation process with a special reference to steroids. Okay, steroids means it's a type of organic compound and it is contain a characteristic arrangement of 4 cycloalkene rings that join to each other. So you know that the basic structure of steroids, you know, it is a cyclopentophenandro ring. It is having a 4 ring is there, and steroids, what is the therapeutic use of these steroids means, you know that for rheumatoid arthritis, allergies, for all type of inflammatory diseases, it is usually used as a contraceptive, then hormonal insufficiencies. Okay, so steroid is also one of the major products that can be used or that can be produced by biotransformation reactions. A lot of steroidal products can be produced by using biotransformation reactions. So what is biotransformation reactions? Biotransformation reactions are those reactions which are taking place in the presence of microorganisms. Microorganisms we are using in order to convert a substrate into a product. Okay. So here see, in this chart you can see the production of different types of steroids. All these are steroids. Diosdenine, sigmasterol, progesterone, hydrocortisone, prednisolone, everything all are steroids. In this chart you can see that some steroids is produced by the chemical means, and some conversions are usually takes place by the in the presence of microorganisms. So it's a mix. So steroids are usually prepared by using both chemical and biotransformation reactions. Okay, for example, diosdenine. Diosdenine can be converted into 11-deoxycortisol. We require a chemical process. And this 11-deoxycortisol can be converted directly to a cortisol or hydrocortisone by using Curvillarial annata. It is a microorganism. Okay, so in order to convert dioxin into hydrocortisone, we have used a one chemical step and one biological step. In the chemical step, dioxin is converted into a dihydroxycortisol. From the deoxycortisol, it is converted into cortisol with the help of one microorganism. Okay, same like that, stigmasterol. Stigmasterol can be converted into progesterone by means of some chemical reactions. From the progesterone, in the presence of a bacteria, Rhizopus nigricans, we will get 11 hydroxy progesterones. From that, by means of some chemical reactions, we can get hydrocortisone and finally cortisone. Then from the hydrocortisone, by using microorganisms, again a biological process, Corynebacterium simplex, you will get prednisolone. From cortisone also, you will get prednisone by Corynebacterium simplex. So don't get confused with the prednisolone and prednisone. From hydrocortisone, in the presence of Corynebacterium bacteria, you will get prednisolone, and from cortisone, in the presence of Corynebacterium simplex, you will get prednisone. Okay, so in this chart, you can see that both chemical and biological processes are usually used for the production of steroids. Okay, so this is the nutshell of for the production of steroids by biotransformation reaction. Okay, so the center portion is progesterone. Okay, from this progesterone, if you see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 products can be used by using different types of microorganisms from a single product or single substrate progesterone. By using different types of microorganisms, we will get 12 different types of steroids. So that is the one of the most advantage of this microbial blood transformation reaction. Okay, just see progesterone. First of all, if you want testosterone, we can add aspergillus or penicillin species. Then progesterones can be converted into 6-beta hydroxy androstenedione by using Gilocladium clatunatum. Okay. Then in the presence of Streptomyces species, you can convert into androstenedione 317 dione. Like that. In the presence of actinomyces species, you can convert into 16 alpha hydroxy pregnane. Then in the presence of macrodiols, we can convert into 14 alpha hydroxyprogesterone. Like that, by using different microorganisms, you will get different types of steroidal products from a single parent model. Okay, so that by using, so while comparing it with the chemical process, it is very difficult to get such type of things. Okay, so coming to the applications of biotransformation reactions. So here you can see that Corynebacterium simplex dehydroxylates cortisone to produce prednisone, and dihydrogenation of hydrocortisone or cortisol to produce prednisolone. Then Nocardia rusticulus biotransform colistin for 19-hydroxy-3-on to esterone. Androstenedione is converted into testosterone by yeast. Peterson and his associates have demonstrated that compounds like progesterone and resisting compounds may be biotransformed to testosterone through the action of Aspergillus flavus and Penicillium adamantici. Okay, so a lot of applications are there for biotransformation in the field of steroids even. Okay, so that's all about microbial biotransformation reactions. Okay, thank you.